Good afternoon, everyone. It's my pleasure to be able to highlight Avrobio to you today. We are a public company, so the disclaimer statement stands. So our mission is curing rare disease in a single dose. And we have a lot of respect for all of the enzyme replacement therapy companies who really revolutionized the treatment of lysosomal storage disorders many years ago. But we also see that gene therapy can be revolutionary as the next step for treatments for lysosomal storage disorders. We're a clinical stage biotech company. We're developing first-line gene therapies for lysosomal storage disorders. And our initial programs include programs for Fabry disease, Gaucher disease, cystinosis, and Pompe. We have compelling Fabry phase 1 data. We've entered the clinic in our phase 2 Fabry program. We treated our first patient in that program earlier this year. And we also are moving forward with our Gaucher program, for which we recently announced that we had a no objection letter from Health Canada to move that program into the clinic. We are targeting standard of care therapies that offer approximately $4 billion of worldwide revenues. The company was founded by 3 of our scientific founders, Jeff Mackay, Kim Warren, and Chris Mason. Several of them are here at the meeting. The 3 of them bring many, many years of expertise in the cell and gene therapy space. And then the team was rounded out with expertise in rare disease drug development. So people like Deanna Peterson, who is also here, working in business development in the rare disease space for many years, myself and Catherine McNaughton, our Senior VP of Portfolio and Program Management. You can see on the right-hand side, I won't read it, but a very highly successful group of board of directors as well as investors. So we have a proven platform, ex vivo lentiviral gene therapy, with proven disease targets. So lysosomal storage disorders have well understood biology. ERT has validated the exogenous enzyme delivery, and there are established regulatory pathways for which we will be following. Lentiviral gene therapy, as you all well know in this audience, has a proven success as well as good safety. So as I mentioned, a lot of respect for enzyme replacement therapy. However, the pharmacokinetics of enzyme replacement therapy are not ideal. What you see on the left-hand side is the pharmacokinetics of ERT, where you deliver an infusion, and that has a very short plasma half-life. In fact, it's gone within the first day after infusion. The patients are then left with approximately 12 to 13 days of no plasma enzyme activity. With our platform, we are able to provide a 24/7 continuous activity of enzyme in the plasma, and we see that as the potential for really improving the unmet need that is left in diseases like Fabry disease. Although ERT has certainly helped those patients still continue to have everyday signs and symptoms like pain, like hypohidrosis. In fact, recently we had Jack Johnson, who is one of the leaders of FSIG, the Fabry Support and Information Group, visit our company. And Jack reported to us, as we have seen in market research as well, that between enzyme infusions, some patients report that their symptoms are starting to come back, the pain starts to come back. And so again, we see this 24/7 continuous production of enzyme via our ex vivo platform as a true positive from the standpoint of our ability to help the ongoing unmet need in these patients. This audience certainly does not need a lecture on ex vivo lentiviral gene therapy, so I won't spend much time on this slide or the next slide, but just to make sure any newbies in the room understand, ex vivo lentiviral gene therapy, we mobilize the stem cells into the peripheral blood. We collect the patient's peripheral blood, and then the blood goes to the manufacturing center. where CD34 positive cells are selected out, transduced with the lentiviral gene therapy, and then cryopreserved. The patient then receives a conditioning regimen, and the cells are infused into the patient. At that time, those cells then hone to the bone marrow where they engraft. They do two things. They produce their progeny, which continue to produce stem cells, as well as producing daughter cells that go on to produce all of the hematopoietic lineages that you see listed on the slide here. Those then produce the enzyme of choice, depending on the disease that we are targeting, and that enzyme is released into the blood and taken up by tissues by a mechanism called cross-correction. In addition, I'll point out for our Gaucher program, in fact, the cells that are actually implemented in Gaucher disease are macrophages, and so those are one of the cell lineages that will be directly corrected by our ex vivo lentiviral gene therapy. Let me spend a few minutes on our pipeline. I mentioned earlier our Fabry program is the most advanced of our programs. The Fabry program consists of 2 clinical trials. The first clinical trial is a phase 1 academic-sponsored study that's being run by our academic partners in Canada, the FACS team. There are 3 clinical trial sites in that program, and they have treated 3 patients, and I'll share some of the data from the program in a couple of slides. The second is our company-sponsored phase 2 study, the FAB 201 study. Now, this study is an efficacy and safety study. The patients are ERT naive, such that this will be the first study that we're able to show the ongoing impact of our gene therapy from an efficacy standpoint. The patients are ERT naive, and the primary endpoint is at 1 year, and we will be looking at reduction of substrate in kidney biopsy. Secondary endpoints consist of many of the functional endpoints that one would expect to see in any Fabry trial. Our next program is our Gaucher program, and as I mentioned, we will be moving into the clinic in 2019 with this program, and we have clearance with Health Canada to do so. Our PAMPA program is earlier in preclinical development, and the PAMPA program, as many of you know, PAMPA is a very difficult to treat disease. I would say probably ERT has done a little for those patients, but it's been very difficult to get enzyme into the muscle, which is what is impacted in PAMPA. We are utilizing a GILT tag or a lysosomal uptake tag with this program to increase uptake of the gene therapy into muscle. And so we're in preclinical development with this program. And then finally, our cystinosis program. This is a program that is partnered with Stephanie Cherqui out of UCSD and GenStem. That program is moving towards the clinic as well, and we have announced that we expect that that program will be in the clinic in 2019. That will be an academic-sponsored study that Dr. Cherqui and the UCSD partners are running as a phase 1-2 trial. So let me just share with you a quick snapshot of the data. I can't share everything today because of the length of the talk. But what I'm showing here is data from the phase 1 academic-sponsored study. On the y-axis, you see alpha-galactosidase enzyme activity, and on the x-axis, time since treatment with our gene therapy AVR-RD01. Patient number 1 is in blue, and patient number 2 is in black. We have data for patient number 1 out to 18 months. And as you can see in the light gray shaded bar, patients with classic Fabry disease would be diagnosed if their plasma alpha-gal levels are less than 1 nanomole per hour per mil in this assay. And so at 18 months, this patient is well above that range, such that if we were to test him not knowing his Fabry history, we would not diagnose the patient with Fabry disease. Now, importantly, the FACS team at the point of the 18-month alpha-gal data were so enthusiastic about the fact that the patient has these robust alpha-gal data that they have taken him off of ERT, and we will now be able to follow his process being off ERT post-gene therapy treatment. In addition, patient number 2, we have data out to 6 months, as you can see, following a similar trajectory as what we've seen in patient number 1. Since we have the patient number 1 data, the academic team has amended the protocol such that patients can be taken off of their ERT therapy at 6 months or later if they're seeing appropriate alpha-gal data. So we will look forward to their decision about patient number 2 being taken off of ERT as well. A third patient has been treated in the trial. He was only treated not long ago, so we don't have data to share on him yet. And I think I also mentioned we've treated our first patient in the phase 2 clinical trial earlier this summer, and we're also seeing good alpha-gal activity in that patient as well. So just a quick touch on our Reg strategy, because it's a little different than what we might all anticipate from those of us who've been working in drug development. So given the fact that we had the phase 1 clinical trial approved in Canada, we have followed that playbook and moved our programs into Canada. Australia uses Canada as a reference country, so we've been able to get approval for our program in Fabry in Australia as well. And then we will move into other territories like the United States as well as Japan. In addition, in rare disease, we always look towards where the patients are. That's an important part of being able to recruit these trials. So, for example, our Gaucher trial will also be run in Israel. And finally, we have designed all of our clinical trials thus far with the end game in mind. So with all of the early approval processes that are available to us that many of the speakers have already mentioned today, like RMAT, PRIME, et cetera, we have designed our clinical trials in such a way that we will be well poised to have further dialogue with the agencies on these approval pathways. Just touch quickly on manufacturing. We are a global company and we have manufacturing in place globally. So you can see, I won't read them all to you, but both plasmid, vector, and cell manufacturing capabilities across the globe, led by Kim Warren, our head of operations. We're well capitalized. We just had a successful IPO earlier this year. And I'll just end with a couple of summary points. We're very excited about the potential to cure rare diseases like these lysosomal storage diseases in a single dose. We are a clinical stage biotech company already in phase 2 with our Fabry program and poised to move 2 more programs into the clinic next year. And our broad pipeline will continue to support ongoing development. So thank you for your time today.